In this episode, we are honored to speak with Professor Asim Surani from the Gordon Institute, a pioneer in germline biology and epigenetic reprogramming. His seminal research has uncovered the mechanisms by which the mammalian germline is specified and how epigenetic information is globally erased and re-established to reset totipotency. How are transgenerational epigenetic marks regulated at the onset of life? What safeguards ensure genomic integrity during this critical window? Join us as we delve into the epigenetic resetting that underpins human development and heredity.
Welcome to the Epigenetics podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Today I'm happy to welcome Azim Surani on this show. Azim, thank you for joining and having the time.
It's a pleasure to be with you.
Yeah, I was talking to Petra Heikoba some weeks ago, and she suggested to reach out to you directly, and I happily did this, and now I'm very happy to talk to you and discover what you've been up to for the last almost 50 years, I guess. So a question I like to start with and to ease into the topic is, how did you become interested in biology in the first place and then in pursuing a career in science?
Well, this is a very long story because as you know, I've been through a lot. So I was actually born in Kenya, and I got very interested in fish and butterflies. There were cichlid fish in Lake Victoria, and there were different colors and things. And there were a lot of butterflies as well. So I didn't know anything about biology. It just seemed fascinating to me, and I had a very good biology teacher who was kind of inspired me to look at the natural world and go and collect samples from ponds and look under microscopes. I got kind of interested generally in that, but I was born in very small towns. I didn't have any role models. I didn't know that you could have a career in science even. I didn't know anybody who had been to a university even. So something that I thought would be interesting to just curiosity, I guess. Yes.
So, since you didn't have any academic background, what would you say to people these days who don't have an academic background and maybe that might be encouraged to do something or thinking about something like go to university or even aim for a career in science?
I think the key thing is to, if you're interested in something and you find something that you think could be fascinating, you just have to follow up on what interests you. I think you come across quite a lot of hurdles that you have to not be deterred by that. I think it's a question of whether you have found some passion for something, natural world or whatever it is, music or mathematics or anything like that. You just have to follow what you think you would like to do, and just keep going. I don't think there's a simple formula. I think there are individual different tastes, and they just have to decide what really interests them. Because the key thing is, are you interested in something? And if you are, I think the key thing is to just follow and keep doing things, and you will find your way through that.
Yeah, I guess you would also gravitate naturally again to the things that interests you, right? So you probably won't go away and do something else. So let's come to your science. Since there are more than 300 papers with your name on it on PubMed, I try to summarize and go through your work on a higher level. So please let me know if I missed something important along the way. I want to start in 1984. There you co-discovered genomic imprinting. Can you walk us through that pivotal experiment and how you got into studying this mechanism back in the days?
Yeah, actually, after I came to Cambridge, I was a student of Bob Edwards, and he was working on in vitro fertilization, human in vitro fertilization. But there were also a lot of people who were working on early mammalian development, which was at a very early stage. There were a lot of pioneering work that was going on. And amongst other things, I met Matt Kaufman, who was working on parthenogenesis. And this was quite a hot topic at that time because we were wondering why you can't have mammals develop parthenogenetically, because you have some vertebrates, like fish and frogs, lizards, they can do that. So the question was, can mammals actually develop parthenogenetically? So that was something which is not my main project. Actually, I was working on blastocyst implantation, but this was the thing that really I became very curious about. I spent a lot of time working on the side on this.
Mm-hmm.
We did lots of experiments, and I was absolutely convinced that I would get a parthenogenetic mouse one day. And it never worked. So then one theory was very prominent at that time, which is called cytoplasmic theory. And what was thought was that at fertilization, the sperm might introduce some kind of cytoplasmic factor into the egg. And this kind of factor was important for development. So if you don't have an egg which is fertilized normally, then you can't get development because of this kind of mysterious substance that was missing. So we did a very simple experiment. Actually, in 1983, we made a triploid with 2 maternal and 1 paternal genome. We found that if you left 2 maternal genomes, we couldn't get development to term. But if you pulled out 1 female nucleus and left a male and a female, then that would go to term. So there was already something that was a kind of a precursor to the paper we did in 1984. And actually, when I wrote this paper for Science, which was published, I put all these ideas about, the kind of imprinting idea. And the reviewers thought this was all very speculative, and they asked me to take it out. So the paper was published with all these things taken out. But then we then the proper experiment, which the question was, do you need both parental genomes for normal development in mammals? And so we made 3 genetically similar zygotes with 2 maternal, 2 paternal, and 1 was normal. And it was only the normal one that developed to term. So that actually told that there was some kind of non-genetic contribution, which is important for mammalian development. And I think the most crucial thing was that we found that we found opposite phenotypes. If we had bimaternal zygotes, we had better development of the embryo with poor placenta. And vice versa, if you had paternal genomes, we had much better trophoblasts. So this kind of opposite phenotypes was the striking thing. So it was clear that there was some kind of non-genetic information being transmitted by the parental germlines. But the term epigenetics was completely unknown or relatively unknown at that time. And I didn't know how to describe this phenomenon. That's why I used the term genomic imprinting, which kind of, to my mind, explain what I was seeing. One reviewer actually took exception to the use of the term imprinting, because he said that there is imprinting in birds, this behavioral imprinting in birds.
Uh-huh.
And but the Nature editor, then I wrote to the editor and said, well, I can't think of any other term. This actually describes what this is telling us. So we were able to use this term at that time.
So how did you then approach to get down to the mechanism? Because obviously you were then probably thinking of what is causing all this, right?
Yeah, so I think it was. So we started thinking about, what could be the mechanism be for this phenomenon, and we started thinking about DNA methylation because it fitted all the criteria. It had to be something that could regulate gene expression, had to be reversible, and and you can reintroduce this kind of a mark. So this criteria actually suggested that DNA methylation could be the imprint. And one of the experiments we did, I think 2 years after the 1984 paper was actually done by Wolf Reich, who was in my lab at that time, which is looking at some transgenes, and we found a transgene which behaved like an imprinted gene. So when it came from the mother, the transgene was methylated. When it went through the paternal germline, it came undermethylated. So this reversibility idea and relation mark being the imprint actually started to take hold, actually. And so we thought that, that we had some pieces there which could explain the mechanism of imprinting.
Mm. So how did you probe DNA methylation at that time?
Oh, we just used this enzyme. There was some enzyme which will cut.
Oh yeah, mm-hmm.
Which is MSP1 and HAPA2, I think. And we were using this restriction enzymes just to check when the transgene was methylated or unmethylated. So that was the technology at that time, and it gave very clear-cut results, reversibility and so on, and the methylation dependent on parental origin. So I think kind of is the idea. And fortunately, there were some other groups who were coming up who had done similar experiments and they also found some of their transgenes were showing this. So I think at that time we calculated that 1 in every 7 transgenes showed this kind of imprinting-like effect. So it was not a rare thing. It was quite common.
Mm.
And after that, we were kind of started to think about endogenous genes, because to get this kind of functional differences between parental genomes, you would have to have some kind of genes that are also working in this way, the real endogenous genes.
So the trans, I just want to get on the transgenes again. So you used this in one study to demonstrate this effect, but you also set up the transgenes to use it as a screen, right? Where you could put transgene into different genetic contexts and then probe for the activity. Was that the same thing that you just mentioned?
No, we didn't. We didn't use transgenes for screening. Actually, what we decided, well, there were 2 things that were happening. One was that Bruce Kattanak was doing some experiments. He was doing genetic experiments, and he was finding that there were some chromosomal regions which behaved in imprinted ways. So if this chromosomal region was duplicated in the maternal, it was maternally duplicated, it gave us a phenotype compared to when it was paternally duplicated. So that was already telling us that there were some regions, chromosomal regions, endogenous chromosomal regions, which were behaving in this kind of imprinting fashion.
Uh-huh.
And so the experiment that we did was actually with Bruce Kattanak, which were also done by other groups as well. So we were looking at this distal chromosome 7, mouse chromosome 7, where you have this H19 IGF2 genes. And so then there was quite a lot of momentum building up. There were other groups that are coming in. And so then all of this work was mutually supportive of the idea that there was something that had, that there was some information coming from parental germline. So we started to look at this region, IGF2H19, and many other groups were doing that too. I think the key thing that came out from collectively was that there was this region that came to be known as imprinting control region, ICR, and which contained the epigenetic as DNA methylation mark, which was introduced in this case, it was introduced when the region came from the maternal germline and was unmethylated, came from the paternal germline. And that kind of explained the reciprocal expression of H19, which was expressed only when it came from the maternal germline, and IGF2, which was expressed only when it came from the paternal germline. So it started to kind of fall together.
Mm.
And then there were other people looking at other regions. Dennis Barlow was one of the key people who found IGF2 receptor was imprinted with the maternal copy expressed, paternal repressed. So, things were building up progressively. But the thing that we started to do for the screen was to take parthenogenetic and androgenetic embryos, and we made cDNA libraries and did a differential screen of that. This was work by Fumi and Tomo Ishino, who were in my lab briefly, and then they went to Japan and continued with this screen. And they found quite a number of genes, PEGs and MEGs, maternally expressed genes. So more and more genes were coming out, and we started to see that there was actually a mark, and it was put in that particular region. So I think this started to tell us something about the mechanisms.
Yeah, I guess that's now the first question would be, how many of those genes are there and are maybe discovered up until now?
I think the numbers do vary. Well, let's say it's approximately 200, something like that. It's around 0.01% of the genome is printed. But the thing that has become quite clear is that these are not conserved.
Mm-hmm.
So if you compare mouse and imprinted genes, only 60% of those genes are conserved. So it's quite a large number, but it's not all of them. So we can talk about it later, but I think this comes back to thinking about the evolution of genomic imprinting, which is something I'm interested in. There's still some mystery about this. So it's still a phenomenon in search of an explanation.
Mm-hmm.
So, which we don't quite understand all of it yet.
It's a very complicated process in the end.
Mm.
So you then also tried to go to the bottom of the mechanism, right? And for this, you also used mouse germ cells and maybe also human cell lines. So how did you approach this? And which factors did you maybe find?
Well, what happened was that, as we and others started to find the genes, and we could see, how these domains were working. And there's a lot of work that was going on. Some of these domains were very large. And I think that was quite important in terms of progress in epigenetics generally, because you can see how these domains can work with a single epigenetic mark. And also the work on human diseases also was starting to come out. But I started to think about this, and I thought that some of the answers must be in the germline, because the process has to happen in the germline. And so I started to think about working on the mammalian germline. It was quite an interesting problem, because I moved to the Gurdon Institute, and I was surrounded by people working on C. elegans and Drosophila and so on. And I would go to the tea room, and people were talking about the germline and things like that. And I realized that, there were big gaps in the understanding of mammalian germline. So we didn't know what were the key genetic regulators of specifications of mammalian germline. It's unknown. So in around, I think it's around just before the year 2000, we started to think about trying to look at the genetic basis for the specification of mammalian germline. And veterinary scientists joined my lab. And this is quite technically a tough problem at the time, because there are only about 30 founder primordial germ cells in mice. And so we had to kind of start using single cell approach. That was the only way we could do it. So I talked to Michinori about this, and we kind of came up with the idea that we could cut out a fragment where we thought the germ cells were, the founder germ cells were, a fragment of about 300 cells. We isolated them, and we calculated that 30 of those cells in that region should be primordial germ cells.
So 10%.
10%. And so Michinori started to make single cell cDNAs. I had my very long-term assistant, Sheila Barton, was quite instrumental in this, because she did quite a lot of the embryology type work. So started making the single cell cDNAs and set up differential screen. Actually, the funny part of this is that I had written a grant application to Wellcome Trust, a project grant application to work on germ cells, because I'd been working on imprinting and I didn't have money for germ cell work. And I talked about this and said, we want to do it like this. We're going to make single cell cDNAs. And actually got the worst reviews ever. And the reviewer said, you're never going to find anything. You only get cell cycle genes. And so I had the worst reviews ever. But the panel at Wellcome Trust thought that this is an interesting idea and somebody should try it. So they gave us a grant, and we started working on it. And it worked quite quickly then. And the first gene we found, which we called Stella, which is very PGC specific. And then we started finding other genes. And the really key gene was the PRDM1, LIMP1, which turned out to be the key gene involved in the specification of primordial germ cells. And then when Michinori went back to Japan, he started working and trying to find other genes, and he found PRDM14. Then it turned out that you need 3 factors, PRDM1, PRDM14, and TFAP2C, to induce primordial germ cells. So we got the genetic basis for primordial germ cell specification. And what this network does is that it represses somatic program. It induces germ cell program. And the important thing from the imprinting perspective was that it also initiates the epigenetic program. So this network actually has starts to have an impact on development of downstream of primordial germ cells. So to give you an example, one of the genes that is repressed by this network is UHRF1, which is important for the maintenance of DNA methylation. And this is repressed by this network. And so we started, and when Petra Haikua joined the lab, we started working on this. And this was Petra did amazing amount of work on this. And so we started looking at what was happening to primordial germ cells as they started proliferating and migrating to the developing gonads. And so this gene, UHRF1, is quite important because since it represses UHRF1, maintenance methylation, as you get DNA replication, you get dilution of 5-methyl cytosine. So this was a passive mechanism for for DNA demethylation. So this is one of the examples. And amongst this, and what we were looking at, what Petra was looking at was erasure of imprints. And indeed, as they went through and as they got into the developing gonads, it reached a point where all the imprints were erased and all the other DNA methylation was erased. So it's called epigenetic ground state because the levels of methylation go down to phenomenally low levels.
Mhm.
It's quite an event.
So did you also look at other chromatin changes? So other factors? Yeah.
We did do that. I think we were, at that time we were mainly focusing on DNA methylation, but I think we were looking at histone modification, lysine 9 methylation, histone H3 lysine 9 methylation. This was part of the changes that we were observing. But later on, when we got into the human germline, we started to also look at both DNA methylation and histone modifications. And in fact, we have published some papers over the last couple of years on histone modification in the human germline. There are some differences compared to mouse, but overall, the system looks similar to what happens in mice.
Yeah, maybe we can come back to this or circle back to this in a few minutes. And because you also looked further into the DNA methylation or demethylation dynamics and imprinting erasure. So what is the mechanism behind the DNA demethylation?
So one of the things that started to also come up was that there could be a role for hydroxymethylation, and the TET enzymes were discovered during this period. And so there was this question of, could hydroxymethylation be involved? And so Jamie Hackett in the lab was working on this, and we did find that there was involvement of hydroxymethylation as well. So this was also contributing to DNA demethylation. Now, I think it's more recently, I think in the context of the human, which we can talk about later, but in the context of the human germline, the view I have is that there are two things that are happening. One is that you have this passive DNA demethylation through repression of VHRF1. And with the TET enzymes, this is, I think, is targeted more at specific loci. So, because what we have to remember is that the germ cell program still continues, despite the fact that you are erasing DNA methylation in the background, you still have to maintain progression of the germline. And some of the genes have to come on at the right time. So some of the later genes, like DASL, for example, are switched on. They're not switched on as a result of passive DNA demethylation. My view is that some of these are this locus-specific mechanism, which is through the involvement of TET enzymes, and that's something that is involved. And I think it's more from the recent work on human germline that personally I have become more convinced about. It's two independent mechanisms operating.
So the general global hypomethylation and the specific TET enzyme dependent local erasure of the DNA methylation?
Yeah. Yes.
Okay. So you already alluded to this. You compared the process of resetting of the epigenome in mouse and humans, right? So maybe you can talk about the key differences that you found.
Yeah, so I think about 10 years ago, we started looking at the human germline, and when Naoko Irie, especially Naoko Irie and Wolfred Tang actually joined the lab, and we were starting to think about the human germline. We thought it'd be very similar, identical to the mouse germline, but we thought nevertheless we should look at it. And there's actually, embryologically, there's a big difference between human and mouse, because the mouse embryos develop as egg cylinders, whereas most of the rest of the mammals, including human, they develop as bilaminar disc during gastrulation. And so we were naively thinking that, it would be just the same, but it turned out to be different. So it turned out to be a tougher problem than we had envisaged. So it took nearly 3 or 4 years to work out the regulators of human primordial germ cell specification. And what was the big surprise, and we couldn't believe it at the beginning, was that it turned out that SOC17 is the key regulator of human primordial germ cell specification. And it was a surprise because SOC17 is a classical specifier of endoderm. So we did genetic. So the thing we had to do is we couldn't do this work on human embryos directly, because although we could get human pre-implantation embryos, there's this 14-day rule, which means that we can't really go beyond a certain point. And it turns out that human primordial germ cells are specified around day 14. So,
Okay.
So that was not feasible option. So we then had to develop in vitro models using human embryonic stem cells. So that's what Naoko Irie did. And then we did genetic experiments using this model and found that, that SOC17 was indeed a key player in this. So it alters many of the things that happen with the specification, also the downstream things on what happened later on. So again, we have these two systems operating. We have this passive DNA demethylation, but then there's also the involvement of hydroxymethylation as well. And we don't actually know at the moment everything about this process between specification and epigenetic resetting. So in human, it starts around day 14. And by the time the germ cells get into the developing gonads, it's around week 5 or 6. So it's a very long, drawn-out process. So one of the things that we found more recently is that one of the experiments that Naoko Irie did, she found that if you induce migration or migration status into the primordial germ cells, human primordial germ cells, you see expression of a gene called DMRT1 and CDH5. DMRT1 is classically known for its role in sex determination, in male sex determination, but it also seems to be expressed much earlier in the human germline during migration. And looking at ex vivo primordial germ cells, we found that it's expressed around week 4 or 5 while they're still migrating. So what Naoko found was that if you express DMRT1 in human primordial germ cells, you induce hydroxymethylation. And what she found also, a key experiment she found, was that if you co-express SOC17 and DMRT1, you get expression of a gene which is called DAZL. DAZL is a key gene which is expressed when germ cells are approaching the developing gonads. It's one of the key genes. It's a kind of a marker for progression of the human primordial germ cells. And what we found with this system was that that DMRT1 And SOC17 targets some of the genes that are expressed later on, including DARZL and some of the genome defense genes as well. So because the genome is getting demethylated, so there are some genome defense genes that are expressed. So these are some of the targets of these two factors. So we started to see that some of the later genes indeed do use hydroxymethylation, and the targeting to those loci, we think, is through SOC17 and DMRT1 working together, targeting certain specific loci. I think this area still needs more work, but I think we have kind of a broad picture of what might be going on.
So you also looked at NANOG, right? What does this do?
Oh, NANOG. So in the mouse germline, we had a very strange result that we could induce specification of primordial germ cells by NANOG. That's kind of a separate story, and that was also a surprise. And I haven't actually thought about it since then. But certainly, what happens in the human germline is that at the specification we have a slight reduction in the pluripotency genes, like OCT4 and NANOG and so on. But shortly after specification, you start to see very high expression of NANOG and OCT4. These are quite important pluripotency factors which are part of the germline, and I think they're an important part of this network. So we call this a reset switch. So the reset switch, in the factors that are involved are SOC17, PRDM1, and TFAP2C. These are three factors. And together with NANOG and OCT4, we call this the reset switch. And we think that this reset switch is the one which induces germ cell program with specification of primordial germ cells, repression of somatic fate, initiation of the epigenetic program. And Wilfred Tang actually published a very nice paper looking at all these factors and how they work. It's a very detailed study that he did.
So next to H3K9 trimethylation, were there other histone marks involved or that you looked at?
We have actually looked at, in the context of human, we have looked at histone H3 TriK27, the polycomb mark, and it does actually come up. I'm not actually, I can't remember exactly. We haven't done a lot of work on that.
Okay, yeah.
So I can't remember exactly, but I think one of the things I should mention is that in the human, when we're looking, and also in mouse, we did find loci that are resistant to reprogramming. And so in humans, some of the genes that are resistant to reprogramming are olfactory receptors. There are many olfactory receptors, I don't know, hundreds or maybe 1,000 olfactory receptors, and it's only one receptor that is expressed per olfactory neuron. And what we found was that these olfactory receptors are not completely reprogrammed. They may lose DNA methylation, but they retain histone H3 TriK methylation and also sometimes the polycomb mark. So there's silencing maintained of these olfactory receptors. And this actually makes sense because these are they are inherited in a silent state. And in the olfactory receptor, one of them, per olfactory neuron, is activated. There are some very elegant experiments showing how these olfactory receptors are chosen, one receptor per a neuron, olfactory neuron. So it kind of makes sense that these olfactory receptors are not reprogrammed completely. They are not reset because they're reset later on in the olfactory neurons. And there are other examples which are also not completely reprogrammed. I mean, amongst them is a transposable element called RTR12C. RTR12C also doesn't get completely reprogrammed. It's a hominoid-specific transposable element, and it may have played quite an important role in during evolution. That I actually was interested in at one point, but I can't remember now all the details.
That's fine. So you also said that by comparing mouse and humans, you are looking at the evolution, right, of these processes. Did you discover something there?
Well, I wouldn't say we're looking at evolution per se. I don't think we kind of went into that kind of depth. But the thing that we were intrigued by was this embryological difference between mouse and human, X-cylinder versus bilaminar disc. And so I've been following some of the work. Actually, what we did was we did do some work on pigs with a colleague, Ramiro Arberio from Nottingham University, because pig embryos develop quite similarly to human embryos. And we worked on pig for two reasons. One was I wanted to actually see whether an actual embryo, not a stem cell model, actually works in the way that I think the human works. And what we found with the pig was that we also see involvement of SOC17. So that network is conserved between human and pigs. And then when people have looked at non-human primates and other mammals, including rabbits, also develop its bilaminar disc. We did a little bit of work on that and found, again, SOC17 is used there. And there's the paper published on rhino, and there we see also SOC17. So it looks like the rodents are the exception. Most of the mammals develop as a bilaminar disc, and they use SOC17. So that's that mammalian system that's used, except for rodents, and they develop slightly differently. But I think we need to think more about the evolutionary implications of this.
So, what are you currently working on? You still have a lab, right? And are you still writing grants? At what might your plans for the next 5 years be?
I don't have a 5-year plan anymore. I have a day-to-day plan right now. So yeah, I have a small lab. I don't know. I have about 4 or 5 people, and we have been one of the things we've been focusing on is to think about the origin of the human germline, because although we have kind of stem cell models and so on, we don't actually have a very good view of how the human germ cells actually originate. So this is more kind of embryological work, which is challenging because we can't use human embryos. So we have to make more models. So we have made new models, looking at like gastruloids and so on. We published a paper recently on that. So we're gaining some more understanding of the precursors of primordial germ cells. And the interesting thing we found was that the precursors primordial germ cells in human give rise to both the amnion and the primordial germ cells. So it's kind of an interesting bifurcation of a progenitor cells. And then one other study which is ongoing by Geraldine Jowett is on looking at slightly later stages of what happens to primordial germ cells. They start off as being bipotential initially. There's no distinction between male and female primordial germ cells, but then later on they start to diverge and become more sex-specific, when they start going towards the female or the male, and they lose this bipotential status. So that's something that Geraldine is working on. So those are the actual experiments that are ongoing, but I alluded to the question of the evolution of genomic imprinting, which I'm not doing any experiments, it's more kind of thinking about it. And I think it's kind of the right time to do this. And the reason is that we know quite a lot about the mechanism of imprinting now. And there's more data which is showing that imprinting originated around about 165 million years ago in the Syrian lineage, in Therians, and which split into marsupials and eutherians. And there are three extant mammalian species, the monotremes, marsupials, and eutherians. And the monotremes, which are the egg-laying mammals, don't have imprinting, but marsupials and eutherians have. So when the eutherians and marsupials split, imprinting was already in place. And so the other thing that is interesting now is that people have started working on imprinting in marsupials. They have been working for some years, but there's more data coming out now. And what we now see is that there is imprinting in marsupials, but there are many differences in the genes that are imprinted. So there's differences in genes that are imprinted in marsupials and also in eutherian mammals. Although there are some canonical imprinted genes which are present in most mammals, including marsupials. So try to putting all these things together, trying to make sense of how the process evolved. And this is a project still in progress. I have many questions, but not many answers yet. I think there are some things that are kind of interesting. So one of the things that I'm kind of thinking about is that there is a cost to imprinting, because there are about 14 diseases that are attributed to imprinting, because you've discarded biallelic expression and adopted monoallelic expression. There's a cost to this. The cost is 14 diseases. But there have to be benefits, for evolution to select this process. So what are the benefits? And so again, these are not very well formulated ideas, because I'm still thinking about this. But if you look at the very early development, what you see is that if you look at the monotreme eggs, they are very large yolky eggs, like what you see in reptiles, reptilian eggs. They're large yolky eggs. You come to marsupials, they have some cytoplasmic inclusion, but they don't have the yolkiness of the monotremes. And then when you come to eutherian mammals, you don't have any of these things around. The key difference between marsupial and eutherians is that marsupials have a unilaminar blastocyst, it's a single layer, whereas the eutherians have a multi-layer. You have an inner cell mass and you have the outer tetraftodome. And if you go back to the original experiment, 1984 experiment, what we found was that the maternal genome in mice is more important for the development of the inner cell mass, or the embryo. Paternal genome relatively more important for the trophoblast. So I'm kind of thinking whether this changes in the embryological development in the tree mammal and the origin of imprinting somehow have coincided somewhere and has played an important role, especially in Eutherian mammals. Because Eutherian mammals are known for two very important things. One is that they have a very regulative early development. So there's a lot of self-selection. So this is an important facet of Eutherian mammals development. And the second is that they can have very long gestation period as well. Now, was this an advantage? So about 65 million years ago, there was the dinosaurs were kind of wiped out. And then you had adaptive radiation of mammals. You see it in mammals. And the fossil records show that there have been many more Eutherian mammals than marsupials. And so I'm kind of wondering whether imprinting might have been an advantage there, whether it played a role in allowing Eutherians to develop some sort of a very regulative development, discarding embryos which are not fit for development to term. And also with other kinds of advantages as well, the long gestation period, so giving the potential for diversity of form and so on. So this is the kind of things that I'm kind of thinking about, but there's still lots to think about.
Yeah, and experiments to plan. So in the last 51 minutes, we've been on a journey through your scientific career, and I tried to get the most important things. Obviously, we missed many things. Would you like to add something to this?
Well, the only thing I could say is that, because people ask me, how did we get into imprinting? We didn't start by the idea of thinking about imprinting at all. It was a very simple idea was, you require both parental genomes for development. And the answer is yes. And I think what came out of that was the fact that I think it was quite important for the field of epigenetics, because, at that time when we discovered the phenomenon of imprinting, there was work on DNA methylation. And so I think this was an important moment in the field of epigenetics, because you had a very important epigenetic mark like DNA methylation, and you had this phenomenon, which was clearly epigenetic. We call it genomic imprinting because we didn't have the word epigenetics at that time. And even as I described in our work, the two things came together, DNA methylation and genomic imprinting. And I think together, this was an important landmark for the field of epigenetics.
But what did those discoveries mean for you personally?
Well, I think the most exciting thing was finding these opposite phenotypes, which I still remember the day we kind of opened up mice and saw this. And we didn't have a mechanism or anything. We only had this embryological studies, but that struck me as a key moment. And I think that kind of opened up an adventure in my career, in my life, actually, because it's sort of, then we got into a lot of other things, started working on the germline and so on. And also along the way, we also did the very first single cell RNA sequencing as well. So a lot of things came out of this very simple idea. And I think, so when students sometimes talk to me, I say to them, if you have an idea, you have to have an idea what you want to try and work on. And then sometimes you find something quite unusual. If you find some exceptional result, don't throw it out.
Pursue
it. Could
it.
be very important. Exceptions can be very important.
Yeah.
So if you go with preconceived ideas, you sometimes discard things which don't fit with what you think how things should work.
So I guess this is a beautiful place to end this interview. So thank you, Azim, for your time and for being on the show.
Thank you, Stefan. It's been fun chatting.
Thanks for listening to this episode of the Epigenetics podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.